Hello everyone, and welcome to our Ask Me Anything session on antibiotic resistance. I'm thrilled to have you all join us today. As the name suggests, this session is your unique opportunity to ask our distinguished guest absolutely anything about their work, research, or innovations in their field. Before we dive in though, a few important details. This is a live session, and we're excited to see so many participants. Many of you submitted your questions when you registered, and we greatly appreciate your enthusiasm. If you haven't submitted your questions yet, don't worry, there's still time. You can enter your questions into the box on the right-hand side of your screen and click Send. Our team in the virtual backstage will ensure that all questions are appropriate, so please keep them relevant. We'll do our best to address as many questions as possible before the session ends. Also, be sure to check out the literature section on the platform for some fantastic resources available for free. And if you encounter any technical issues, click the chat icon in the bottom right corner and our team will assist you. Now, once again, welcome to our Ask Me Anything session. Without further ado, it is my pleasure to introduce our expert for today, Siegwart.
Hi.
Hello, Ed. I think we can get started today with the usual career questions, and I'm sure that could set us off to begin with. So what initially sparked your interest in microbiology and the field of antibiotic resistance?
Oh, that's a good question to start with. So I started my microbiology journey, if you like, with a company called Britvic Soft Drinks. I actually did work experience from school in a microbiology laboratory, doing environmental monitoring, testing Pepsi and various orange juice products and things like that. So then I kind of got the bug. And from that point onwards, went and did it at university, did a pure microbiology degree at university, left and joined a hospital lab to gain some experience, some work experience down in Southampton. And then from that point on, I joined a company called GR Micro, who were a contract research company who focused on antimicrobial drug development. And from that point on, I spent the next 10 to 15 years of my career working for a CRO that got acquired by a number of different company names, working with lots of other people in the sector, developing antibiotics and learning from what they were doing, really. And then 5 years ago, I joined Oppilotech, who are one of those drug development companies I did contract work for. And that's where I am today.
Fantastic. So it's always been microbiology for you since the beginning, really?
Yeah, I'm that kind of sad nerd who only has one track mind with microbiology, and it's since I got into working in antimicrobials as well, that's kind of just been my focus, and I've been quite stubborn about it. It's a bizarre thing. Ideally, I think in science you want to try and stay flexible, but I've done exactly the opposite. I've said I know I'm only doing antimicrobials, and that's the thing for me.
And within this passion for antimicrobials and the miniature world, is there anything that really drew you in your undergraduate years or any time around there?
Well, antibiotics in of itself is quite a compelling thing to want to go after, because that these critical things that underpin medical systems and something we critically undervalue as a society, but really desperately need to make new ones of. And I think that's what drew me to anti-infectives, because when I joined my first company working in anti-infectives in 2007, the industry was waning, and it's continued to wane since then. But the need for those antimicrobials has not changed at all. We still desperately need new ones making. So that's what attracted me to it, really.
Of course, important reasons, and I'm sure we'll get on to discussing those in great detail as we go on. But before we get to those urgent matters, I'm curious, when you began at Oppilotech, what's been the journey like within the organization to become where you are now, I believe, Director of Microbiology?
Sure. So it's a small company. It's founded by four friends who are the CSO, the CEO, and then the Director of MedChem, and one other who's since left and joined other organisations. But yeah, it's a very small company. There's only 6 of us who work there currently. So we all join and we all immediately get fancy job titles like Director of Microbiology. It's not a lot of other companies in anti-infective development are like this. They're very small. They're not massive teams. You don't rise through the ranks and gain position. You come in at those positions, but you don't get to join those companies without experience. You need to have evolved that experience over time. And I like to think I did my time working in the CRO world, battling for quite a number of other organizations to get them the data they need to get to where I am, really.
I'm sure you weren't just handed the title.
You never know.
I'm curious, what is it about the sector that lends itself to these smaller companies then?
So the big companies kind of left and it kind of abandoned the sector. There's a handful of large pharma organizations that still operate in the sector, but they do so out of the necessity for making novel antimicrobials. They don't do so because there's profit in it. There's a massive issue within the industrial sector in pharmaceuticals, in antimicrobials, because there's a delinked cost between the societal value and the development cost. So what it means to society is a lot more than what we're allowed to charge for developing them. And that means the two don't marry up. You end up having to produce a drug which costs millions and hundreds of millions, if not billions of pounds, to then get to the market and not be able to charge that money back. So no one will invest in it. And that's kind of part of the reason why AMR is such a big issue, that so many companies, the large organizations, don't really operate in the space anymore. A lot of them have ducked out, and the ones that do are doing some great work, but they're not as well backed as they would have been in, say, other areas like oncology or, analgesics, etc. Those guys are a much more investable and a much more attractive opportunity for those larger organizations.
Yeah, that's an unfortunate disparity between the funding and the need. I'm sure we'll get into that a bit more, but we do have a question pre-sent in by an audience member that's a little bit more generic, get away from the specific industry stuff for a bit, and back to yourself.
Yeah.
How has the field of microbiology changed since you started working in it?
Yeah. Well, it's changed personally because I've gone in and out of the lab quite a few times. I'm currently still working in the lab. But when I first started in the hospital labs back down in Southampton General Hospital, we were still doing everything the old school way. So microbiology is basically culture from swabs off a patient. You identify the bugs that are on there through a various range of biochemical tests and different media and things like that. And then you identify what the pathogen is, and you tell the clinician, and you inform the clinician what antibiotics they should use to treat the patient. What's changed now is it's much more an automated kind of thing. Things like the moldy toff have come in and changed the way you identify those bacteria. It's not so much based on the skill of the people working on the lab as much. These people are still there, but it's more based on more advanced techniques for identifying the bacteria. And I also think the situation's changed with antibiotic resistance. It's obviously much worse now than it was when I first started in the industry, and the lack of options for doctors is obviously very, very concerning.
Has that changed, the latter change that you highlighted, the increase in antibiotic resistance, has that, it's hard to pick a phrase here, but affected the mood of people who work in this subsector? It affected the incentive of the work?
I guess so. I think that there has been a certain brain drain, particularly on people working in anti-infectives. I mean, it's something that regularly gets brought up these days, that most of the people who work in the sector are talented enough to go and earn money doing other things in other sectors. So that there's definitely an element of that. And I think the people who work in clinical ID labs, there's also a brain drain there. So the microbiology labs where I started, they're not as well staffed as they used to be, and the kind of the fun's been sucked out of it. I used to enjoy identifying the bacteria. Like I say, I'm a massive nerd, but I used to enjoy identifying the bacteria and going through the process and doing all the biochemical tests. Whereas these days, you literally just put it on a little target, put it in a machine, and it tells you what it is. So it's not as exciting as it once was, perhaps.
I thought that innovation and that experience is something that makes your job faster. has taken a little bit of the fun out of it.
Yeah, a little bit. As a microbiologist, it has, but by the same token, it's a lot more accurate, and the data you're giving, especially in the patient doctor capacity, is much more accurate, which is obviously more important than me having a good time.
Good. And that links into another question we've had sent in, which is a little bit more on the lighter side. What has been your most significant research breakthrough in antibiotic resistance so far?
Cool. As far as individual things go, I think joining Oppilotech has obviously been a big breakthrough for me. I think to jump from being on a contract research capacity to working for a company that's actually developing their own antimicrobials was a big thing for me, and it was something I was really keen to do. But I remember before I joined Oppilotech, seeing the CSOs speak at a conference and seeing what they were doing and their approach to drug development in this field was something that was uniquely different. So I was really glad it all panned out into a job working with those guys.
And that's what drew you into Oppilotech then?
Yeah. I mean, I wind the CEO up. I say, it's a lack of other good options. And there are a limited amount of jobs in the space. But yeah, no, Oppilotech have a genuinely innovative approach to antibiotic drug development that nobody else is doing. And I think innovation in this field is something that's vital. That if we're not doing something different, if we're just doing exactly the same as what others have done previously, then we're not going to get the results we need.
Fantastic. It's good to hear the focus on new ideas. I'm sure we'll hear more about those ideas in a bit, but I can go to another question we've had sent in, and that concerns the practical aspects of your life, I suppose. Can you walk us through a typical day in your lab, and what are you currently working on?
So this week I have been doing, we do client-based services as well. So part of a lag from my old career is we also offer service work in the lab. So I've been doing some service work this week for a couple of clients. So there's been some MIC testing there. So MIC is minimum inhibitory concentration testing. You're basically inoculating a test drug against test bacteria and seeing which bacteria the drugs work against best. So there's been a lot of that. Then there's been our own testing. Our lead program is currently in hit to lead development. So I've been testing novel compounds that we've had generated by a chemist, CRO chemist, who sent those new molecules to us. And then I run them again against the bacteria and tell our med chem in our team whether they're working or not. And then we can use that to identify things like structural relationships with the activity of the drugs. So yeah, that's the typical week. I spend most of my time in the lab. I mean, last week I was at a conference. I was speaking at a conference, which was a really cool thing to do for me personally. But yeah, most of the time you're sitting in the lab plowing through the work. But this is the world of a small company. You have to do lots of different things. It's all networking, lab work, and then you do some of the more executive functions, things like filling out forms for grant applications and things like that as well.
Interesting. And speaking of the nitty-gritty, we have some questions that concern some more practical issues within the world of lab work. So I've got one here. What are the main steps involved in preclinical antibiotic drug discovery?
Oh, how long have you got? So you basically first want to identify a drug target, something you want to go after. If you are going down a target-based approach, you can also go with a target-agnostic approach, where you're literally just testing things like naturally occurring compounds against a range of bacteria, and then you can do the other way around, identify what target they're hitting. But our approach is mostly target-based. We identify a target within the bacterial biochemistry that if we can inhibit that target, we kill the bacteria. And then we develop drugs that will do exactly that, which will kill the bacteria. And then you have to go through a range of other tests, identifying how that drug is going about inhibiting that bacteria, if it's doing by hitting your target like you think it is. And then you can go on from there, developing the molecule further, increasing its specificity for the target, reducing things like toxicities, because you're obviously putting it into a person. You don't want to kill the person. You want to be able to kill the bug. And so you need to make sure you bleed out the toxicity while at the same time keeping your activity against the bacteria. And then from there, you test in animal models, you do your PK/PD, you do your ADME, and this is where lots of acronyms come in for the uninitiated. And then you work through those tests to then establish what you call a clinical candidate or a lead candidate, I suppose. And then you go into lead optimization, improving the functionality of your molecule, and then on to do first in human trials for safety. And way on from that, you develop the drug on from there.
Not a short process then.
No, it's really not, and it's a challenging one in anti-infectives because it's low budget, it's low funded, and a lot of the time we're small companies, we don't have these in-house services to ourselves. We can't do in vivo testing ourselves, so we need to outsource it to a third-party CRO. And they are also operating for all the other sectors, the other pharma sectors, who aren't struggling financially and can pay higher prices. So there's kind of a disconnect there where we're trying to keep up with the rest of the pharma industry, but we haven't got the same funds. So, yeah, that causes issues too.
We had one question submitted in that I was going to maybe leave it until a bit later towards the end, but I think this is a good time to include it as we've discussed some of the issues that stain the sector of antimicrobial resistance. Would you have any advice for a researcher young in their career who is considering going into the field?
I mean, yeah, the head says don't. The heart says it's important and you absolutely need to do it. I'd say academia is probably your best start. It's the most stable way of getting into the industry. Find yourself one of the institutions in the world who are paragons of antimicrobial drug discovery, and they do exist. There are definitely I'm not going to list them now because I'll forget one and then they'll be upset with me, but find one of these universities, study there, find somebody there who you can study under and work with them and learn that way. And that's a really good way as well. If you later then want to spin out into industry, that then gives you that option. That's what I'd do. If I was a young, keen microbiologist wanting to get into anti-infectives now, I'd look for those academic institutions and try and get in there.
Oh, good advice there for the young researchers we have in the audience. Got many more questions, more specific to your function and role in antibiotic resistance, so we can get onto those. What role does systems biology play in identifying novel targets for antibiotic therapies?
Oh, crikey. So, yeah, the biology of cells, you're basically, when you inhibit a cell's function with a drug, you're basically stopping some of the biochemical functions of that cell. That's very much in our approach. We are mapping the biochemistry because we want to understand how in impacting that one target within the biochemistry will lead to either cell death or inhibition of growth. So obviously understanding all the biochemistry and the systems biology involved in that is vitally important. And it's something that not a lot of people have done within, at least in the anti-infective industry today, because a lot of drugs that are even currently on the market, their mode of action and the way they inhibit bacteria is not fully understood. There's still people publishing information on, say, penicillin. There's still people publishing new modes of action on that very recently. So yeah, understanding exactly what your drug is going to do to a cell is obviously really useful information if you're then going to go and develop that drug further.
Of course. And we've discussed the need to fight antibiotic resistance, huge crux of your work. But we've had a question just come in now, actually, from someone in the audience about how to reduce antibiotic resistance in a productive way. Any sustainable alternatives?
Sure. Well, there's loads of ways. The first way is prevent infection. Things like wash protocols, things like making sure people are washing hands, people are practicing good sanitation, prevent those infections wherever you can. You can also conduct surveillance, so you identify where the worst antimicrobial resistance outbreaks are occurring, so you can try and contain them so they don't spread. Access to pharmaceuticals in areas where the global burden of disease is highest. this is another question that could take about 4 or 5 hours to answer properly. But yeah, there's lots you can do on the preventative side before you introduce the agents, and that also then spares those agents, because every time you use an antibiotic, you're increasing the chance that resistance is going to develop against it. So anything you can do to prevent an infection is fantastic, and we should keep doing that and encourage others to do so. And there's also a weird disconnect with members of the public that we don't properly have a good understanding of infectious disease. I think with COVID happening recently, you can see how some members of the public just can't quite get their head around infectious disease and how it works. And that understanding that better and educating people better on their understanding of that is going to help prevent things like AMR, because it will prevent infection spreading.
Of course, it's good to hear the dual strategy being stressed there, not just addressing antibiotic resistance, but prevention as well.
Yeah.
And along the same thoughts, we've got a question just submitted now, which I suppose may have been overlooked towards the beginning. It's setting the scene for this whole conversation, really. What are the main reasons for antibiotic resistance, antimicrobial resistance?
Crikey. Well, it's going to happen anyway. It's evolution, basically. It's the purest form of evolution you're ever going to see. That literally, I can go in the lab tomorrow and start subculturing a bacterial strain in with an antibiotic and keep subculturing it, and eventually it will evolve resistance. And the way it does it is there are billions of individual bacteria within a population in each of those test cells. And one of them just needs to be a mutant that has a mutation that confers resistance to that drug. And then that one will outgrow the rest because there's no competition anymore. And then there's other things like resistance mechanisms that can jump from other organisms in the environment. They can move between bacterial organisms. So not all bugs are susceptible to all antibiotics. Some bugs have resistance profiles but are not pathogenic. So they're not harmful to humans, but they may be carrying resistance gene elements within their genome. And if they transfer to a pathogenic bacteria, then you've got evolution of resistance through that way as well. And yeah, anything we can do to reduce antibiotic use is going to help keep our armamentarium of antibiotics going. But antibiotic resistance is inevitable. It is going to happen regardless of what we do.
Really sets the grand stage of your work and the work of others like you, really. It's fighting nature in this grand sense.
Yeah. There's an element of working with nature. There are approaches that do that. There's microbiome-based therapies and things like that that try to work with the natural activity of the bacteria and try and prevent them just hurting humans as opposed to just straight up killing them. So yeah, there's lots of approaches that go around it that way, but yeah, it's inevitable. AMR is something that's always going to happen.
Worthwhile to keep in mind. We've got another question coming from the same member of the audience who asked the question about alternatives or how to reduce antibiotic resistance. So they ask again, in public health, how can we guide the local public about this? Something you discussed towards the end of your response about really educating the average person so we can all help together.
Yeah, it's difficult because it's not necessarily something people want to hear about. They don't want to hear about being ill or being sick or things like that because, yeah, it's not a nice thing to talk about. But I think education starts in schools. I honestly think I remember back to being in school and not learning anything about infectious disease until I got really to just before university. I remember I did A-levels. I'm that old. Back when I did them, I had to beg my tutor to add a module on microbiology. And he said he wouldn't do it because there wasn't enough interest in it. And things like that are kind of seeded in kids early. So I think making sure that the general population are educated right about what infectious disease is and understand how it spreads and how dangerous it could be, it is a really good starting point. Education's vital.
Gotta start young.
Yeah, exactly. How we do it is not easy because the general population now are of an age where they don't necessarily want to learn anymore. I think, again, using COVID as an example, when people laid out the facts of COVID, scientifically in an educational way. People weren't interested. They were either scared or they were just going to do their own thing. And it was, yeah, it's a very difficult thing to educate a population. But I think the best place to start is in the education system. That's something that needs to be at the core of it.
Yeah. Good words to go by. I'm going to switch track to some of the more specific practical questions, again, going back and forth between some of the pre-submitted questions and some of the live ones we've got coming in.
Sure.
But someone did ask, what advances in technology have had the most significant impact on preclinical drug discovery for antibiotics?
Ooh. There's quite a lot of things that have gone on, but it doesn't necessarily bleed through to the sector. I mean, things like genomic sequencing has obviously made a big difference. I'm a bit funny about genomic sequencing, though, because genomic sequencing will only tell you what pieces are on the board within a bacterial genome. It's not going to tell you how those pieces are going to move. So things like biochemical modeling, bringing our own work into it again, kind of gives you a bit more information on what's going on, but you obviously can't do that for every cell you're going to test. But yeah, I'd say genomic whole genome sequencing has made a big difference because you can identify things like resistance genes and use that to infer mechanisms of resistance, which then helps with things like surveillance and keeping track of where the resistance is and the organisms that it's operating in. And it also guides you in development because you're identifying those genes that are most dangerous in pathogenic bacteria in the clinic, and then you can then develop against those bugs, making sure that you're tackling what needs to be tackled, where the clinical need is.
Know your enemy.
Yeah, definitely.
Got another question. Can you talk about any exciting developments or new approaches currently being researched at Oppilotech?
I like to think they're all exciting, but I think, yeah, our lead program, which is slowly and steadily moving forward, is a novel target, and the fact that we understand what inhibition of that target does to a cell, and we know it's a good drug target, means that if we can get a successful inhibitor up and running and into the clinic, we will have a really good novel drug on our hands. And I think once we've done that and we've proven and validated that that methodology works, we can do it again, and we can do it again, and we can do it again. Because it's the sort of thing, once you've modeled everything in a cell, you know what's good and what's not. And you've got a list of good drug targets you can go after, which is kind of our shtick, really.
That's good to hear. And how do we address the challenges posed by multi-drug resistant MDR bacteria?
So, yeah, again, they're something that are inevitable. The more we use antibiotics, the more MDR bugs are going to occur. There's a particularly nasty strain of Klebsiella rotating at the moment that's hypervirulent and highly resistant. You need novel therapies to treat them. Not necessarily small molecule therapeutics like we're developing, but there's all kinds of other things like vaccines, phage, peptides, and then you need all those preventative measures that I mentioned earlier as well. Anything you can do to stop the patient getting ill with those bugs in the first place is going to help. You've just got to develop new things. That's the bottom line.
Yeah. Keep going in the race. We can talk more about, I suppose, the differing states of antibiotic resistance across the world. We've had a few questions that concern that pre-submitted. So how does antibiotic resistance differ across different regions in the world? What challenges do low middle income countries face, perhaps?
Okay. Yeah. Well, the global burden of disease is largely in lower middle income countries. Places like Africa and sometimes India is thrown in where that group as well. These places are where the burden of disease is. And the organisms that cause these infections aren't necessarily what the West are developing drugs for. So that's something we definitely need to factor in when we're making development decisions. Lower middle income countries are a tricky one because obviously they don't have the money to reimburse development costs because they're low income countries. But then nor do the high-income countries at the moment with the current model of reimbursement for development of antimicrobials. So you could argue there's been examples of things like public-private partnerships that have made drugs available to people in lower middle income countries that otherwise wouldn't have had access to these drugs. And in tackling the global burden of disease in those areas, you also tackle the opportunity for resistance to spread from those areas to others. And a lot of the time, problems in independent areas are restricted to certain types of organisms or certain types of resistance. So, for instance, in the US, they still very much feel when you talk to clinicians that there's a big MRSA problem over there. Whereas in Europe and Asia and even Africa, MRSA doesn't even factor into the decision-making process. There's other things like TB is obviously a massive problem in Africa. There's the Klebsiella I mentioned, which is a big problem in India. Carbapenemase resistant Acinetobacter is a massive problem across Asia. So there's different bugs that cause different problems elsewhere. And you need to factor that in when you are making development decisions. So for a small company like us, you've got quite a lot to consider when you're deciding where to develop your drugs. But you need to factor in the fact that there are different resistance profiles and different bugs causing different problems all over the world. I think that the conditions that those organisms cause can also be a factor as well, because Staph aureus can cause like skin and soft tissue infections. It can cause pneumonia, whereas Acinetobacter and other organisms like that tend to be more pneumonia based, but they can also cause other conditions. And it gets very, very complicated very, very fast.
It may sound obvious, but the data that informs your work, the work of your company and others like it, at least in Europe and North America, that data really is coming from further afield. So to be properly informed, you really have to know what's going on in other parts of the world.
Surveillance is a major issue in antimicrobials at the moment. I used to do quite a lot of antimicrobial surveillance in my previous role. And you can see how certain resistance and certain bugs occurs more frequently in some areas, but you need to understand where that resistance is, particularly where you're developing your drug as well. If you intend to market a drug, say in China, you need to make sure you're hitting the bugs that are causing problems in China and not the ones that are a problem in the US.
Of course. And looking beyond the work of companies that you work for, like Oppilotech and research, what role do international regulatory bodies such as the WHO, CDC, play in shaping research priorities for antibiotic resistance?
So the WHO, for instance, the World Health Organization, published critical pathogen lists, as does the CDC. So they say what the problem organisms are. The CDC can have a slightly more US focus because obviously the US CDC, but whereas the World Health Organization obviously focuses on where the global burden of disease is. But the organisms that they list and the research priorities they give out tend to be the same. And the World Health Organization do quite cool things where they publish target product profiles. They're saying we're looking for a drug that does this, and this to tackle this disease in this area, which as a developer is really, really helpful, because the only other way you're going to find out that is by contacting someone who works in a clinical capacity in that area of the world. And for a small company like us, that's just not going to happen. But they both play quite a fundamental role because they guide where we target development. And obviously that's going to affect what drugs we create inevitably.
Of course, you mentioned the importance of global surveillance. And when it comes to global surveillance of disease and maladies, I don't think there's any organization that reaches as far as the WHO. So,
they do. The WHO operate something called GLASS, which is something that collects a lot of surveillance data from other sources and then encourages other areas where surveillance is patchy to upscale their laboratory operations so they can provide that kind of data. But there's still massive gaps in our understanding of where the resistance and where the bugs that are causing it are coming from.
Of course. We've got a question that is adjacent to antibiotics, I suppose. Are there any promising non-antibiotic therapies, such as immune-modulating agents, that could play a major role in future treatments?
Yes. Short answer. Yeah, no, there are. So definitely vaccines have a role to play. There's certain bugs you can't develop vaccines for that are going to be a massive developmental headache to develop for. But there are some bugs that stick out as excellent opportunities to develop vaccines against. There are already bacterial strains that vaccines exist for, things like the Hib vaccine, strep pneumo vaccine. These are things that are already widely used and do have an excellent effect on reducing infection levels. But there's also things like phage, which has excellent opportunity for use in chronic conditions. I think things like cystic fibrosis, recurring UTIs, things like that, where phage might have its best impact. And then there's lots of other antivirulence modulators and things like that, that instead of tackling the bug by killing it, they turn off its pathogenicity, if you like. They stop it being something that will cause an infection, because obviously humans are full of bacteria already. So just turn those nasty pathogenic bacteria into something a lot less dangerous for the host.
And speaking of danger to the host, we've got a few questions pre-submitted that concern the illnesses that we've really been discussing treatment for. And one just submitted now, actually. How to recover an antibiotic-resistant human body in a sustainable way without harming the body. So I appreciate that your medical expertise may not extend into clinical doctor-approved advice, but I suppose the question concerns treating or how any doctor and medical professional may go about treating someone who is dealing with antibiotic-resistant bacteria.
Sure. So I have a personal story, I guess. I had bronchitis quite a number of years ago now. It was maybe about 12, 15 years ago. Where because I worked in a microbiology lab at the time, I managed to culture the bug that was causing my bronchitis and tested it against a range of antimicrobials. And I found it was resistant to almost everything. It had high level resistance to almost everything. And I went to the doctor and I explained the situation. She was like, "What? How did you test your..." But once we got past the part where she realized I was a microbiologist, she basically said, "Well, I can't give you anything to treat your infection." And basically, I had to just get better over time. And normally, you go to a doctor with these kinds of infections, you have like a course of antibiotics, and in 2-3 days, you feel pretty much well again. And then you finish the course and it doesn't come back. But it took me 3 months to get over that bronchitis without any antibiotics. And that's basically the future of what we're facing with resistant infections. So other ways of dealing with that, prevention is the best one. If there was a way, and again, personally speaking, I gave myself bronchitis because I was ill with a cold and I carried on working in a microbiology lab, which was a really stupid thing to do. But these infections you can get just from not looking after yourself when you're ill. So prevention is obviously something that's very, very important in that case. There's lots of other kinds of therapies that are in development that aren't quite in clinical use yet, that aren't specific antimicrobials. Things I've mentioned before, like phage and peptides and vaccines to prevent. And even once someone's infected, there are certain types of vaccine you can give people that help. But yeah, the bottom line is the best thing to do is not get ill in the first place, if you can.
Prevention stressed again. That must have been a difficult 3 months for you, but I can see how it's informed your work since.
Definitely, it's not something I'd wish on anybody. I mean, it's a deeply stressful time because you're ill. You know, you're not circling the drain or anything. You know, you're young enough and you're healthy enough that you will get better over time. But to be ill for 3 months with something that previously would have just been a few days is very frustrating.
And highlights the serious problems that could arise in patients who had compromised immune systems or who were older but in that same situation.
Exactly. Yeah. Anybody who's already not well in some kind of way. If they get those kind of infections, obviously the consequences are much more serious. But yeah, I think I'm keen to express that this is something that affects every single person. That set of circumstances isn't necessarily unique to me. That could have happened to anybody and will happen to people in the future.
And continuing on the rather morbid outlook, we had a precedent question about what role healthcare professionals play in combating antibiotic resistance, particularly in regions with high resistance rates. So this chimes in with what we discussed before about prevention and other ways that antibiotic resistance can be challenged, really.
Yeah, good stewardship. Stewardship's the term that people use to describe appropriate use of antimicrobials, which is easier said than done. I do appreciate that doctors and clinicians and people who treat patients are left with some very difficult decisions in these set of circumstances. I mean, the Hippocratic Oath itself is do no harm. And you've got a drug that you can give to a patient that you think might make them better, but then they might evolve the bug might evolve resistance to that drug in the future and end up hurting a lot more people. So do you restrict use of that drug for that one patient, or do you? Yeah, it's a really difficult set of circumstances that clinicians are facing. And they're basically at the moment going around treating people with cocktails of antibiotics. They're looking for new research for best use of antimicrobials, the right dosage for the right patient. Things like diagnostics are very important as well, identifying exactly what the right bug is so they can get the right drug to treat the right patient. Things like that are very important to clinicians. But yeah, it's a difficult job that's only going to get worse. Yeah.
Do you have any data or insights that those kind of healthcare practices on specifically identifying the bacteria and sourcing a particular antibiotic for it rather than a broad spectrum one? Are there any areas of the world or any case studies where that has, in the real world, proven to be a success?
I can't think of any specific examples off the top of my head, but yes, certainly that is ideally the way a lot of people want to go. They want narrow spectrum agents that specifically target certain bugs, but you've got to have the solid diagnostic in place to identify exactly what that bug is before you treat a patient. Because say you get a patient in a ward with meningitis, that could be fatal in hours, if not days. And you need to treat them immediately. So you haven't got time to run a diagnostic. You need to get something in them quick. So you're not going to gamble on withholding an antibiotic in that set of circumstances.
Of course. Well, we've only got maybe 15, 20 minutes or so left, and we've moved quite quickly onto the broad scale picture of the world and many countries and the whole state of antibiotic resistance. But in the time we have left, we can hone back in on your own personal work, research, and the work of Oppilotech. So we've got a question here. How is Oppilotech applying AI or machine learning in the early stages of drug discovery for antibiotic resistance?
So yeah, it's all in silico modeling. So all our biochemical modeling we're doing is in models on computers, often using cloud services, because otherwise our computers literally start catching fire. But you basically model the entire biochemistry. You collect information either through wet lab testing of our own. Our CSO has done over a decade of research collecting information on various different stimuli, on various different bacteria with genomic knockdowns or genomic knockouts. So you can turn off and on genes within the bacterial biochemistry, and then you can feed that into a model. You can then improve that model further by identifying a particular phenotypic state or a particular part of the metabolism, and you feed in even more information on detailed things like metabolic rates of enzymes, and it gets really intricate, really detailed information so that you've got everything in that AI model. We've kind of moved away from using AI because it's not really AI, it's not really learning. You teach the original model with a machine learning model, but from that point on, it's not really artificial intelligence. It's not doing anything. You interrogate it with an input and it gives you a response. But yeah, it's all in silico. So it means we don't have to do the 10 years of wet lab research that have come before it, we don't have to do any further wet lab research unless we're trying to improve the model further.
Interesting stuff. And we've got another technical question. What types of assays are used to treat new antibiotics in the preclinical stage?
Treat. Yeah, well, it depends what they're asking there. So most of the time, the assays we use are these minimum inhibitory concentration tests. So there's all kinds of published guidelines. I've literally spent the majority of my career doing these MIC tests, and there's published guidelines on how you do it properly. And there's ways to do it wrong as well, which a lot of people seem to miss, because the test itself is actually quite a simple test. You do a concentration range of your test antibiotic in wells on a microtiter plate. You add in a standard inoculum of your test bacteria at a known concentration, and then you incubate it and see where it grows. And your concentration range you reach a point where the bug stops growing, and you call that concentration the minimum inhibitory concentration. So that's the lowest concentration of a drug required to inhibit growth of that bug. So that's the first kind of assay you do, because obviously if you can inhibit growth of your target bacteria, you're doing something to the bacteria, and that's great. But you can also do more specific biochemical assays. If you know what your target is, for instance, you can literally synthesize that protein target, get hold of your substrate, and then you can add your drug into that reaction and see if it inhibits the function of that protein and its substrate. So there's all kinds of assays you can do in preclinical development, but they're the main two, really.
No, that was a good overview. Very welcome. We have another question. How are preclinical models of infection used to assess the efficacy of new antibiotics?
In vivo, did you say?
I did not. I just said preclinical models of infection.
Preclinical models of infection. So it depends what they're talking about, really. You've got to assess things like PKPD, which is what they might be talking about, which is the pharmacokinetics, pharmacodynamics of your particular development compound in a biological system. So you want to make sure that the biological system itself is not editing your drug to impair its function, but you also want to make sure that your drug is performing its function as you expect in that biochemical system. But there's all kinds of early stage assays you can run that will tell you more information about your drug before you get to that point. And then when you get into PKPD, and this is not an area I consider myself a mega expert in, but yeah, when you get there, you can start working out performance index parameters, pharmacodynamic index parameters, and it gets very complicated and very sciencey very, very fast.
Oh, still some good insights there. Thank you for addressing or working around some of the nuance in that question, perhaps. We've got a few questions pre-sent in that really concern the careers of people in your position and the sector itself, things we've touched on throughout this talk. Mm-hmm. How do you stay motivated? I think there's some implication there about the ongoing challenges and setbacks in the field of antibiotic research.
It's the kind of job where you can look yourself in the mirror at the end of the day and know you're doing something that's good, that's going to save people's lives, that's vitally important, whether or not there's the finance there or the lack of challenges there is neither here nor there. I think there's most of the people I think who are still in this sector now have had to fight tooth and nail to stay in it. So I think that some of the most committed people I know are working in anti-infective drug development right now, and they won't go away from it because there's almost certainly been opportunity for them to do so, and they haven't yet. And I know people who are frequently asking me, have you got any jobs going, who really want to who've previously worked in the sector that can't get back into it because it's such a shrinking pool of potential organizations you can work for. It's you stay motivated really by knowing you're doing something good. And I remember having a conversation in a bar with my CEO recently, and he was saying he's worked on the investment side of things, and he knows people who've made millions, and they're just kind of, yeah, we've got lots of money, but what do I do with it? And I don't know if he was telling me that. It sounds like the sort of thing you'd make up to try and motivate your staff. But if he was telling the truth, I feel really fulfilled doing what I'm doing, and knowing that what I do will fundamentally save people's lives and reduce the suffering of others. So, yeah, there's your motivation right there.
Fantastic, noble stuff. And we've got a couple questions that both look to the future, one in optimistic sense and one in a more pessimistic sense. So the first question is, you mentioned the state of the sector, the shrinking pool of investment, fewer and fewer companies of smaller size. Do you see any change on the horizon, any different model expansion, or are we still shrinking?
So there's good things that have happened. The UK subscription model is a perfect example of that. It's a delinked model where the UK pays for access to 2 new antimicrobials instead of based on volume of sales. So they pay a standard subscription amount. They call it a Netflix model. So you pay for a month of Netflix, you can watch as much films as you like, and it works the same with the antimicrobials. You pay for access to that antimicrobial, you pay a flat fee, and you get access to as much as you need within a certain threshold. And that's something that's world leading. That's something that all countries need a version of in order to reimburse for antimicrobials, because that delinked nature of it means that if you develop an antibiotic, you are guaranteed a payment at the end of that process that covers your development costs. And that then attracts investors back into the space. It means that companies can stay solvent through development. It's a tiny step in the right direction. It's not solving the problem at all. It goes a tiny step towards it. There's a similar act in the US called the Pasteur Act, which is obviously on a much larger scale because it's the US market. That's sitting in Congress, and there's a lot of people who've been working very, very hard over the past few years to try and get that bill passed and into law. But as yet, it hasn't happened. And that's the kind of thing that if that were to happen, there'll be a domino effect of other countries rushing to do the same. Because if the US is playing ball with a subscription model, then the rest of the world has to do the same. Otherwise, they won't get access to the antimicrobials at all. So there's good things that are happening in the space. I mean, literally yesterday, there was a UN General Assembly meeting on AMR, and they've published an 115 point list of things that the world needs to do to tackle AMR, and they're all solid suggestions. And then following up from that, there's a ministerial meeting in November to react to that UN General Assembly meeting outcome. So world leaders will meet and say, well, this is what we're going to do off the back of that. So, yeah, there's good things that are happening in the space. It's not all doom and gloom, but these solutions are long-term. The political solutions are not going to affect anything in the next 5 years. So we kind of need stopgap measures until that happens. That's the bottom line. There's optimism, definitely. And you're always stuck with this weird, am I positive, am I negative about it? Because if you're too negative about it, there's no investor that's going to touch you with a barge pole. But if you're too positive about it, you're not going to instigate any change. So, yeah, you're always juggling both of these. And at the same time, I'm just a scientist. I'm just a guy that works in a lab. But I'm constantly looking at UN General Assembly meeting proceedings, looking for it's a crazy, crazy industry to be operating in. Yeah, definitely.
But it's good to know there are good ideas on the table. Mm-hmm. And it was the US, in a sense, that pioneered the Netflix model. So hopefully they can further adopt it in different areas. We'll see. Yeah. I don't mean to end the Q&A on the pessimistic note, but I did mention that more pessimistic question. Maybe I should have gone the other way around. But we did have a question sent in. Do you ever worry about the post-antibiotic era?
Well, yes. I do, and I don't think people fully appreciate the risk that we're all facing as a society from it. I think that comes from the education point I was saying previously. If people fully appreciated what the risk of a world without antibiotics is, then there would be faster change. Those that do understand it do advocate for it and do push for it. I mean, there was a poll in The Guardian, it must be about 12, 15 years ago now, and Guardian readers are a bit more scientifically minded, shall we say, and they highlighted antibiotics as the number one scientific discovery of the 20th century or something like that. So they get it. People get that antibiotics are vitally important. But yeah, the post-antibiotic era is something that does worry me a lot, and I think it's already here to a degree. Things like surgery is being cancelled because it's not worth the risk. Oncology patients are now dying from infections where the oncology treatments have been made that tackle the cancer, but now they die from infections instead. It's like 1 in 5 cancer patients now gets an antibiotic-resistant infection or something like that. It's an alarming number. But yeah, the impact of a post-antibiotic era is way bigger than people realise. And that's something we need to change.
Mm-hmm. And that can inform your earlier remark about education and can inform earlier remark about motivation. Absolutely. But perhaps it would be better to put your last 2 answers in the other order. There is the optimism out there, but there is also the reason to avoid.
Yeah, there's plenty of innovation in the space. There's some really good science out there, but without the finance to instigate it's going to be a struggle to actually make anything like that a reality. I mean, when Fleming discovered penicillin, he discovered penicillin, but he didn't make penicillin. It wasn't until other people like Florian Chain came along and turned it into a drug that we actually had the drug of penicillin. And that's kind of, we're stuck in the same world where we've got lots of Flemings, but not enough Florian Chains at the moment.
I think that's a good place to leave the Q&A there on, Ed. Hopefully we can get a few more of them from webinars such as this. Some inspiration to be found. So I can say thank you for joining us today, Ed. Unfortunately, we've reached the end of our session due to time constraints, but it has been a pleasure speaking with you. The feedback has been fantastic, and it's clear our audience have enjoyed the discussion. Before we close, we'd love to hear from you, the audience. Did you enjoy this session? Please let us know via the feedback form that will appear shortly on your screen. This AMA will be available on demand, so feel free to share the link with anyone who might be interested. Once again, thank you all for your time and participation, and thank you, Ed.
Thank you. Yeah, just a final point about if people do want to get in touch with me, LinkedIn is the best place. If you've got any questions I didn't answer here, please do reach out and get in touch.
There you go. Thank you again.
Thank you.